In today's class, we will continue with the chapter cellular fate processes. So in previous class, we looked at one fate process, that is cellular differentiation, correct? Now in today's class, we will look at another fate process, that is cell migration. If any one of you can tell me what do you understand by the term cell migration? It means what? Cell is moving from one position to another, correct? So if have you guys seen cells in the microscope and ever observed this phenomena yourself? Anyone familiar with cell culture technique and etc.?
No, ma'am, I have never seen migrating cell under a microscope.
Okay, so usually you cannot actually observe means it happening, means you, because cellular migration is happening at a time scale that is much more different for us to comprehend. But usually what happens, if you mark the point, say it was present at location A, and after some time, say 1 day 2 day, the cell would have moved to another position. Okay, it has left the original position. It means cell has migrated. Okay, so we can actually do some experiments and give a concentration gradient of some attractant molecules, chemoattractant molecules, which will cause the cell migration. Okay, so these kind of experiments we will talk later, but in today's class we will actually understand how cell migrates. Okay, what are the steps a cell has to do in order to move from, say, position A to position B. So if we break down the entire cell migration process, we can do it in 5 steps. So first step, what happens is the membrane lamellipodia, it protrudes, that is protrusion of membrane lamellipodia. So in this figure A, so this is a cell, basically this is a cell, and this is the lamellipodia. And this lamellipodia is but nothing but the cell membrane that has extended out a little bit. Okay, this is the first step. So here let's describe what happens here. So this extension, it happens because of actin polymerization, and that too at a local region. You guys understand that there are molecules like actin molecules and myosin molecules that are present in the cytoskeleton of the cell. Okay, so these actin molecules, they form polymers where like a pearl chain. Okay, and this polymerization, because of this polymerization, this formation of lamellipodia is possible. So how does this polymerization happens? So usually sites to which, or the open sites to which new actin molecules can bind, those sites increase in number. Okay, and then at these sites, at these new sites, new actin monomers, means small, if like a pearl chain, and each bead is a actin monomer. So these actin monomers, they get added to the new sites that is generated. Now at these sites, the polymerization reaction is happening and a actin polymer is formed. Now these sites can arise either due to uncapping of existing filaments. So if this is a chain, say uncapping means few monomers, they have like been taken out. Okay, so new site has generated. Or severing means this pearl chain, you just cut it and open the pearl chain. Like a garland, you open it in order to add a few more ropes. So in that, severing is happening. Or completely new nucleation sites will generate, like de novo, new sites, complete new sites, to which more and more actin monomers will bind and ultimately form a actin polymer chain. And because of this actin polymer chain, this lamellopodia structure, membrane lamellopodia structure is formed by the cell. Now, after it has done so, means it has extended its cell membrane area, correct? It has extended it by making membrane lamellopodia. Then next step is adhering. That lamellopodia has to adhere to the surface, that is a matrix. And this adhesion will happen via receptors called integrin receptors. And these integrin receptors basically provide a link between the substrate. So this is the substrate, okay, and this is the integrin receptors. Sorry, I have drawn it wrong. So as I was saying, this is the substrate and this is the cell, correct? And some receptor like this is coming from the cell surface. So this integrin receptors is acting as a link between the cytoskeleton. Now in the cytoskeleton, I already explained that there are actin polymer chains. So this is the actin polymer chain that is there in the cytoskeleton. So this cytoskeleton is connected to the substrate by the integrin receptors, okay? And this particular type of adhesion or the point of adhesion is called focal adhesions. So because of this formation of membrane lamellopodia, it is then adhering to the substrate, that is the matrix, via receptors called integrin receptors and creating what is called focal adhesion points or focal adhesions that are formed at the interface of cell and the substrate. So various complexes or protein complexes are known that regulate the formation of these focal adhesions, like this Rho subfamily of RAS family of GTP binding proteins. These GTP binding proteins, you can divide the proteins into families, and then within the family there is a subfamily of protein. So this is just for information. So let's go back to this figure. I talked about step 1 where lamellopodia has extended. Then in step 2, this lamellopodia, it has attached, okay, or adhered with the substrate with formation of this kind of focal adhesion points, correct? Now as I said, this is a 5-step process. So we have to cover 3 more steps. So in step 3, what will happen? Here, contraction of cytoplasm takes place. Cytoplasm, entire cytoplasm content of the cell, it contracts, and this contraction happens through the action of myosin proteins. Okay, so you must have heard these terms actin and myosin, contraction and relaxation. They help in contraction and relaxation. Also, when you have studied about how muscles contract and relax in class 12th, so there also we talked about these class of proteins. So one actin filament is there in cytoskeleton, and with the actin filaments is another cytoskeleton protein that is called myosin, and this myosin protein is going to help in contraction. Okay, now because of this contraction effect, okay, the cell is able to move forward, means a traction force is, cell is able to apply a traction force on the substrate. What do you mean by traction force? So you guys must be familiar with the term friction, right? So friction is a force that the substrate gives to the body to resist its motion, correct? Its friction is not going, if the friction is too high, the body is not going to move on the substrate. Whereas the traction force is the force that is applied by the body on the substrate in order to move forward. So the traction force is higher than frictional force, then only the body will move. So cell is doing similar thing. Cell is trying to overcome whatever frictional effects are there the substrate by contracting itself and generate a traction force. Now people have actually measured what is the magnitude of this traction force. So in fibroblast, people have reported that the value of traction force is around 2 raised to the 2 into 10 to the power 4, means around 20,000 microdynes. So this is a unit of force, and this force is around 1,000 times greater than a single myosin motor, means it is 1,000 times greater than what the traction force generated from a single myosin molecule. Okay, now note that the force required to move a cell is dominated by traction forces, correct? अगर traction force ज़्यादा नहीं होगा तो cell will not move, and this force is obviously more than whatever resistive forces are there. So since cells are usually present in a fluid environment, so there will be a drag force provided by the fluid, surrounding fluid. So this cell is trying to move through the fluid, so the traction force is going to be greater than this drag force. Okay, now this is the step third, where you can see the cytoplasm, the content of cytoplasm, they have squeezed up. Okay, and that is because a overall contraction force is being applied by the myosin motors to generate this net squeezed up effect, means a contraction force is there. So this content they have basically squeezed. Okay, initially it was like this, now due to contraction force it has, this content have become squeezed, contracted. Okay, now after contraction what will happen? There will be release of the rear and cell displacement. So if this part of the cell is rear and this is like the front part, so in the fourth step what is happening, this rear is going to release. Okay, it's going to release, and cell has moved this much of distance, going to release and go in the forward direction. Basically the lamellopodia that is there in the front is in the direction of where the cell is trying to move. Okay, so cell is trying to move in this direction, that is why this huge lamellopodia is extended in this direction. Okay, now let's see what happens during the rear release. Now during this rear release, both physical and enzymatic processes have been involved. That actually explains how the cell's rear is getting released from the substrate, and also the adhesion process. Now in this step the adhesion is asymmetric, means a part of the cell is adhered whereas the one part of the cell is not adhered, it is released. So that is why it is asymmetric. So what all physical process contribute to the release? So first is the in the rear, like at the back side of the cell, there are fewer integrin receptors. There are fewer integrin receptors, because of which there is fewer focal adhesion points. So force, that the contraction force that each focal adhesion point is going to experience is going to be much greater, correct? It is going to be distributed, this contraction force is getting distributed among small number of focal adhesion points, which is why each adhesion point is experiencing a greater force. So that is contributing to release of the rear end of the cell. Another physical effect, another physical phenomena that is contributing to the release is the dynamic formation of integrins that have a bias for breaking contacts in the rear. Means whatever focal adhesions that are formed at the rear end of the cell, they are more biased for getting broken up very easily, whereas in the front part it is not so. They are more like biased towards sticking. In the rear end they are more biased that we have to get released from the substrate. Now, enzymatic processes also favor this release of the rear. How? The intracellular concentration of this calcium ion, they also help in breaking the integrin and matrix complexes, that is in breaking the focal adhesion points. The calcium ion content, they are contributing it. So these are, as I said, regulated by some protein complexes. Now I assume that these calcium ions, they must be interacting with these protein complexes, and thereby causing some downstream effect of releasing or breaking the focal adhesion points. Now, in some cell types, people have observed that when the rear is released, some part of the membrane is ripped out. Okay, so what happens? Some integrin receptors are still bound to the substrate, whereas and it has basically integrin receptors. These are what? These are transmembrane proteins. They have ripped the membrane, cell membrane, and the binding strength is so large that it is still uh, adhered to the substrate while the cell has moved forward. So these left behind integrin receptors, they are called retraction fibers. So this is the fourth step, where you can see the focal adhesion points, they are getting released. So if they are as I said, if it is released, this rear end is released, the cell will move, say, this much amount of distance. So you can understand how slowly the cell migration is happening. Okay. The last step, what happens? Now suppose in a cell type where this ripping effect, membrane ripping effect is not happening, all the integrin receptor molecules are still intact and is bound to the cell membrane, then what will happen? Then recycling of this integrin receptor will take place. So the cell is efficient in a way, like do not waste in resources. So what it is doing, it is doing endocytosis to take in all the integrin receptors, okay, inside the cell, and it is getting transported towards the leading edge, that is the front part of the cell. It is getting transported, and due to endocytosis, whatever content is there, it is encapsulated within a vesicle. So this vesicle is transported towards the leading edge. Now when these integrin receptors, that is encapsulated within vesicle, when they reach at the leading edge, there these integrin receptors will again go to the cell membrane, okay, and help in adhesion. All right. Also, newly synthesized, if the cell is supposed simultaneously synthesizing few integrin proteins as well, they are also getting transported to the leading edge. Now you can see that the adhesion at the leading edge, so here the recycling is happening and coming in the leading edge. So the adhesion in the leading edge is more biased, means here the strength of adhesion is much more than the strength of adhesion at the rear end, because in the rear end there is less number of molecules of integrin receptors, and thus less focal adhesion points are getting formed, where in the leading edge at the front of the cell, more adhesion points are getting formed. So in this way, again the same thing will happen. All when cell has finished all these 5 steps, again it will extend, some leading edge will little bit will extend, correct? And then what will happen? Due to extension, this extended part of the cell will adhere with the matrix, correct? After adhering, the cell will generate a contraction force, correct? And due to the effect of this contraction force, the rear end will get released, and the cell has moved some distance forward. After the release, all the integrin molecules that are present at the rear end, they are getting recycled and getting transported in the front end, and this process will repeat and repeat and repeat until cell reaches its destination, okay, where it wants to go. Until it reaches there, this process will continue. So any query in understanding these 5 steps of cell migration? Any doubt? No doubt?
No, ma'am.
Okay. Now we can actually, just like we described differentiation process mathematically using some mathematical models, similarly we can describe the cell migration process as well through 2 approaches. One approach is considering the entire cell population, that is the whole population approach. And another approach is if we track the motion of individual cells. So these are 2 approaches of understanding the understand the dynamics of cell migration mathematically. Now, the whole population approach we will discuss first. This approach is taken to describe the migration of population of cell, and it is very much similar when people describe the diffusion process of a large number of molecules. So, it is analogous to the molecular diffusion process. So I wish to know, like, if any one of you in your normal college curriculum have studied about mass transfer or molecular diffusion. Have any of you guys have studied it?
No, ma'am.
No.
Well,
Okay. So,
personally, I study the process
so you have studied.
a little bit mass transfer. That is in based on the bio process, how much mass will be entering the system and how much goes outside per time, like that.
So it means you're describing a mass balance equation. So you have studied mass transfer as a subject, entire subject. Have you studied?
No, ma'am, no, ma'am.
No. So okay, fine. Thank you. So as I was saying, that mass transfer is basically a chemical engineering course. Okay, that is also very much relevant for biotechnologists as well. And as you can see here in this example, that it is helping, this molecular diffusion process is going to help us in order to describe a very important cellular fate process, that is cell migration. So I'll go through slowly in this part. So if you guys have any difficulty in understanding any equation or etc., you guys let me know. So, so this whole population approach also assumes that all the cells involved, they are not replicating. Okay, so they are not undergoing any cell division. So they are only migrating. Only the migration process is being assumed. And the cell migration process is studied in 2D dimensions, means in 2D. Okay, you take a substrate in which the cells is migrating. So you must remember it is a 2D process. And all the experiments that are used to quantify the cell migration, they are all done on substrates, like on a flat surface. So the number of cells that is migrating across a particular location is described by this equation. Okay, so dx by dx, so this is the a capital X is the cell number and small x is the distance coordinate. So this differential equation is equal to cellular flux. So flux is a quantity that helps us. So you have guys have studied about magnetic flux, electric flux. Like flux basically says that how much of anything, say current or like here in this case cell, is moving. Okay, is moving from like across a particular area and in a unit time. So a time factor is also there while uh, describing flux. Okay, so the rate at which some substance is moving through a area, this is a general definition of flux. But here in this case they have actually not taken the time dimension. Okay, so in this equation you cannot see any uh, term that this is related to time. Here only cell migration or the cell migration of cell across a location is given, means across some distance is given by cellular flux. So don't get confused here, like why flux they are describing without time or etc. like that. So it is different than the normal flux definition that you will find elsewhere. Can any one of you guess why they haven't take the time term here in this to describe the solar flux?
Ma'am, can you repeat why they didn't took time?
Yeah, means why didn't they quantify the migration of the cell across a distance with respect to time?
Because it differs cell to cell.
You are correct.
The moving.
Yeah. So here, this is a whole population approach, right? So each cell in the within a population, so there are say millions of cells. So each cell will like migrate to a distance by taking differing amount of time. You cannot assume that all 1 million cell will migrate within 1 minute to say location B. Okay, not all the cells will reach location B at the same time. That is why the cell term, so the time term is not taken while describing the cellular flux equation. But the distance you can measure. Okay, distance is constant, so you can describe with respect to distance. Now this cellular flux term J is related to cell concentration, means the cell number basically X, and the chemokine concentration. Now you must remember, cell usually doesn't migrate, or means it does migrate. Okay, it can show random walk kind of a phenomena, means just like you guys have heard about Brownian motion of molecules. Like the molecules, they they hit each other and then move anywhere they want, right? So all the molecules, they're hitting each other and moving anywhere. So this kind of random motion is there, but cell also migrates in response to some uh, concentration of chemokines. So chemokines, we will talk, they are soluble signaling factors, and when a cell senses a concentration of this chemokine, then cell migration process is triggered. It is basically a signal for the cell to migrate to a particular target. It tries to migrate to the point where to the point from which the chemokine is being released. So this point or the source of this chemokine could be another cell type. Okay, uh, that cell type is trying to call other cells to participate in some kind of a process. So we have recently studied a similar kind of a process, like where cells are calling each other and doing a particular process. Can you guys guess what process I'm hinting on? So it's wound healing, right? So in wound healing, initially there are platelets, right? So they try to secrete various signaling molecules and try to call other inflammatory cells, and then fibroblasts and the epithelial cells, etc. So this is how, like the real time, this is how a process is happening. And one cellular fate process, that is cell method, that we are talking about, is playing a very important role for the wound healing process to proceed. Okay, so as I was saying, that the cellular flux J, it can be related to cellular concentration and chemokine concentration using this particular equation. Now this J is equal to minus sigma dx by dx. Now this term is called random motility. So random motility is basically like used to describe that cell is showing a random walk kind of a behavior. It is moving anywhere it wants. Okay. Then this term minus x by 2 into d sigma by dA into dA by dx. This term is defining the phenomena of chemokinesis. Basically, how the cell speed is changing with change in the chemokine concentration. Okay, and the last term, this is chi, this is a Greek letter chi into x dA by dx. Now this term is for the chemotaxis. Now chemotaxis, we have already looked into chemotaxis, right, in the previous class. So I described there are various mode of cell migration. So one mode of cell migration is chemotaxis, which happens in presence of a concentration gradient of a chemokine molecule or chemoattractant molecule. Okay, now observe this equation, you will see some coefficients or constants. Now this sigma is one constant, then this chi is another constant, and this a is another important factor in this equation. You know, x is the cell number and small x is the distance coordinate. Now this sigma is the random motility coefficient, chi is the chemotactic coefficient, and a is the concentration of the chemoattractant, basically the concentration of the chemokine. Now we'll talk about each of these phenomena individually. Now this random motility is very much similar to the molecular diffusion process, and because of that, the term that is used to describe random motility, it's very much similar to the mass flux equation that is coming from Fick's first law. So as I was saying, uh, in molecular diffusion, if you go into details of this particular topic that is taught in mass transfer, so here they use a law called Fick's law, okay, to describe how what describe the change in concentration, okay, how the concentration of the solute or the movement, the movement of the solute is happening from higher concentration to lower concentration. So this particular statement is described using this particular equation, where d is the diffusion coefficient and this is the concentration gradient dCA by dx. Ca is the concentration of the solute, and you know, as I said, that molecules or solid molecules, they always move from concentration where there is high concentration to low concentration. So this particular law or this particular equation, the random motility equation, is similar. Okay, and this random motility is basically a measure of the random nature of cell migration, means how the cells is moving randomly. It is that particular effect is explained by this particular term, random motility term. And if there is no chemokine present, or if the concentration of the chemokine is uniform means there is no concentration gradient present, then these two terms, chemokinesis and chemotaxis, will not happen, because of which these two terms will be simply zero, and only the random motility effect will dominate. And all cellular flux, that is the change in cell number, okay, or the migration of the cell across some distance, all that will happen because of the random motion of the cell. Okay, now chemokinesis, it is the measure of change in cell speed with the concentration of the attractant, that is chemoattractant. Now this chemokinesis term here, this term is usually very small. Okay, this term is very small in comparison to the other two terms, that is the random motility and the chemotaxis term, and it is like wise to negligible, like the value of this term is very less, so people usually neglect it in the term. But rest assured, this effect is also happening and is contributing to the overall cellular flux. And the last effect that is contributing is the chemotaxis. Now this term represents the biased movement of the cell in presence of a concentration gradient of chemoattractant. So concentration gradient is there, means there is a high concentration region of chemoattractant and a low concentration region of chemoattractant, and cell is moving after sensing this concentration gradient. And this term again is analogous to convective mass transfer diffusion term. So this is the convective mass transfer diffusion term. Okay, so Kc is the convective mass transfer coefficient and delta C is the concentration gradient. Okay, so this term is similar to this term, where chi here is the chemotactic coefficient and x dA by dx. So this is the dA by dx is the concentration gradient of the chemoattractant, and x multiplied with the cell number. So this term is similar to the convective mass transfer diffusion term. So again, this is coming from the molecular diffusion knowledge. So it is taught there in more detail. And this chi value, the chemotactic coefficient value, is actually difficult to measure, because in experimental condition it is difficult to maintain a stable concentration gradient of your chemoattractant over a few seconds only. Okay, if you are using few microliters of some growth factor, etc., within seconds the concentration will become uniform. You won't be able to get very stable, like a gradient that is maintained for some time period, and within this time period it is you are also have to observe the cell migration process. So it is difficult, but it is not impossible. People have done it by optimizing the conditions. Okay, now the some typical values of sigma and chi. Okay, in order to help you understand like in what like the order of magnitude of the cell flux that is happening. So the sigma value, or the random motility coefficient, people have measured it in various kind of cell types and in vivo microenvironment as well. So this value ranges from 10 raised to the power minus 8 to 10 raised to the power minus 7 centimeter square per second. This is a very, very small value. And if you compare it with the diffusion coefficient terms, this D term that is coming from the Fick's first law of molecular diffusion. Okay, if you compare it with diffusion coefficient term or diffusivity of a small molecule, say like very, very small molecule, like the small अगरबत्ती का जो धुआं निकलता है. Okay, so all the that particular smoke is composed of small particles. So that small I'm talking about. So if you compare it with the diffusion of the small molecules, then that value is around 10 to the power minus 5, whereas for cell it is 10 to the power minus 8 to 10 to the power minus 7. So cell is moving at a very, very the diffusivity, or you can say, is very, very less in comparison to the small molecules. And when you compare it with the protein, then this value is again 10 times smaller in comparison to the diffusivity or the diffusion coefficients of the protein. Whereas people have also measured the value of chi, the chemotactic coefficient, and this value varies with the strength of the concentration gradient of the chemoattractant, and people report it to be in the range of 10 to the power minus 2 to 10 to the power minus 3 centimeter square per molar second. Okay, so this is about describing cellular migration process when you are looking at the whole population of the cell. So any queries so far? Any queries?
No, ma'am.
Okay. Now we can also track individual cells and see how the cell is moving. So the motile cells means the cells that which are moving, they appear to undergo a random walk process. Means they will move anywhere. Wherever they find space, they will move there. Okay. So and it is also possible to track the motion of each cell. And this tracking and all, it is usually done, or the data that is obtained from this tracking, they are all done using a computer. Computer softwares are available, and individual cell tracking analysis when people are doing, they describe this phenomena using terms like cell speed and persistence time. Okay, so this is how a cell migration, if you track one cell, if you track the path of one cell, this is how the path would look like. So this is for the neutrophils, individual neutrophil cell that is undergoing a completely random motion. Okay, random, wherever it is finding space, it is moving there. So if say it is, the cell has started here and it is moving here, then there it is changing directions, it is then going straight for some time, then it is changed direction, again going straight for some time, then change direction. So wherever it is finding space, it is moving there. So it is showing a random kind of a walk, random motion, like Brownian motion of molecules, and people can track it. And when they track it, they can plot this kind of a figures to show the cell migration, individual cell migration. Now persistence time, as I said, people use two terms, persistence time and cell speed, to describe this process. Persistence time is basically the time for which the cell is moving without changing its direction significantly. So if the cell is moving in this direction, for how long it has moved in this direction, that is the persistence time. And then at this point, the cell took a turn and moved upwards. Okay, so again a new persistence time value will come for this particular path. So in this way people are calculating, whereas cell speed is basically the distance by time. You know, speed is the it is given by distance by time. So similarly, it is the average distance that a cell travels per unit time. Simple as that. And this speed, the value of this speed, it depends on the time interval chosen. Correct? So if you monitor or the track the cell motion for say 10 minutes, so for 10 minutes only, you whatever average speed you will get, that is for valid only for the 10 minutes. The next 1 hour, the cell speed will be different. Okay, and if the time for which you are tracking the cell motion is much greater than the persistence time, in that case you can actually come up with this expression, which correlates the random motility coefficient that we talked in previously, right? Just in whole population description. So there we talked about random motility and the random motility coefficient. So here, since the motion of the cell is completely random, the other two effects are negligible here. So we're only taking the random motility effect. So random motility coefficient now can be related with the persistence time and the speed of the cell using this particular expression. Okay. Now here in this table, values of speed and the persistence time that people have measured for different cell types is given. Okay. So that's it from my end. Okay, and we will continue with the chapter in the next class, like tomorrow, where we will discuss about cell division and then cell death. Okay, and then we will finish up with the cellular fate processes. Is there any doubt so far?
Ma'am, today
Yeah.
we discussed about these calculations, right? That is migrating cell speed and some sort of directions, where these concepts will be helpful or applied in tissue engineering, ma'am.
So in particular, see, you are thinking in perspective of only making a tissue construct, all right, but there are
Hmm.
various fundamental questions that people might need to answer to understand the performance of a particular tissue construct. So these
Hmm.
are basically a fundamental question. So if someone asks you that the scaffold that you have made, within that scaffold, how the cell is migrating, this is a very fundamental question. right? Whether there is enough space for the cell to move. So then you can use these cell migration calculations and use the expressions, and then use analysis and then tracking, and then track how the cell is moving around or within the scaffold. So these are
Okay.
fundamental things. Okay.
Got it, man.
Okay. Any other queries? So, okay, I wanted to point out a few things. So in last quiz, like in quiz 3, there was one question. I think question number, wait, question number 4, part B. So that question actually was incorrect. All right. So the question should have been, let me uh, tell the question. So in the paper, I had written, why do you think microencapsulated islet cells, when transplanted subcutaneously with the 3D printed scaffold, didn't reverse diabetes? So this question is incorrect, because when the authors encapsulated these islet cells and then put it inside the 3D scaffold, then only a reversal of diabetes was observed. Individually, these encapsulated islet cells, they couldn't couldn't normalize the blood glucose level. So instead of with, there should have been without. So because this question didn't make any sense, so the overall marking that I've done is out of 14. Okay, 14. The total marks of the paper has turned to be 14 because I have removed this question from the evaluation. But I saw that few of you have written the correct answer, but don't worry. But many of you have not attempted as well. So it is like win-win kind of a thing. I hope you guys can understand this. Also, nobody have actually pointed out during the exam as well, so I would have corrected it during that. No issue. It is a part wrong thing that happened from my end. Actually, I didn't check that question properly. And also another thing, I also marked the assignments, and the second question, question number 5, I wanted to discuss with you guys. Okay, so let me show you that question. So question number second, actually, where you had to calculate the cell numbers, etc. Let me show that question. Okay. Is my screen visible? So this is this question that none of you could do it correctly. So here they actually they have given you two tissue types. One is the skin, another is the cartilage. Now from our class discussion, you already know that cartilage contains less amount of cell. It is mostly ECM, and whereas skin is the very polyproliferative organ. So the cell numbers that is inherently present in cartilage and skin, they will not be same. First thing to note. So many of you have taken the same assumption that both the tissue type will have the same cell number and done the calculation for part A. Okay, so that is again going to be incorrect. So and in the next part, in the part B, here here they have asked that how many cells that you need prior to expanding your cells within the TEM, so that you achieve the therapeutic cell number, that is from million to billion cells, correct? Means the therapeutic cell number that you calculated from part A. This is the part A is giving the therapeutic cell number. So in order to achieve this therapeutic cell number, how many cells you need in the beginning? So here is actually part for solving part B, you have to assume a value, and that assumption should be coming from an educated guess. All right. So if you look at standard cell culture practices, then you will understand, like from a standard T25 flask or standard T75 flask, how many cells are can be obtained. Okay, from if you could have think in that direction, then part B I think would have been correct. Whereas in part C, you had to use the cell growth equation. Do you guys remember the cell growth curve? There is a lag phase, log phase, right? And the stationary. So you can for the
Yes.
so for the exponential phase, that is the log phase, so people have derived equation where the change or the increase in cell number directly proportional to the number of cells that is present at that particular moment. So I have given this calculation completely, but for part C you had to recall that concepts of cell growth equation, etc. I think you guys have studied about it in bioreactor analysis, etc. These kind of courses they teach cell growth, how mathematically they describe cell growth, and then the cell death also they describe using some constants and differential equations, etc. So from that, if you applied that knowledge, you could have easily calculated the time that is going to take to reach the final therapeutic cell number, and this doubling time were also given. So here I have done this calculation, etc. I will share with you all the solution, like maybe tonight or tomorrow. Then you guys can thoroughly look at the solution, and if you guys have any queries still, if you guys still have any queries, you guys can let me know later on. Okay. So let me quickly take attendance, and then we will wrap up today's class. It is 11th. So one by one, tell me your name so that I can mark it.
Janvi.
Janvi. Okay,
Murli,
Janvi.
ma'am.
Okay, Mulli next.
Nidhi Priya.
Nidhi Priya. Next,
Vani ma'am.
Vani. Next,
Kirtan.
Kirtan. Next, sorry, can you repeat?
Shambhavi.
Shambhavi. Okay. Next, anyone else left? I think I have marked for Mulli, Vaishnavi, Shambhavi, Kirtan, Janvi, Vani, and Nidhi Priya. Okay. So that's all. If you guys have any queries or anything you would like to share,